is Dr. Trio. He is an expert in pharmaceutical development, currently serves as CEO and chairman of Oncotelic Incorporation. Previously, he was president and CEO of Igradasol development of second generation Abraxane, where he pioneered the regulatory pathway for approval of paclitaxel nanomedicine through a single bioequivalence trial against Abraxane. When Igdrasol merged with Sorrento Therapeutics, he became CSO and board director. He was board director of Sinomed, a company focusing on central nervous system drug development. And today he will present the topic dealing with TGF-beta as a therapeutic target for COVID-19, results of RT19 and C001 trials. So welcome, and we are waiting for your presentation.
Okay, can you hear me?
Yes, we can hear you.
Okay, so thank you for having me on this meeting. Today I will tell you a little bit about TGF-beta, its role in oncology, as well as role in COVID-19, which is surprising change of indication for TGF-beta. Oncotelic is a TGF-beta company. This is some forwarding statement because we are a publicly traded company. Oncotelic is really a conversion of three company, Ponder AI, where we have the cluster computing capability, where we have a supercomputer that can sit on your desktop, and couple that with blockchain back office support. We feel that this will change how drug development will be performed in the future. Oncotelic brought to the table the antisense platform, focusing on TGF-beta, and Mation have the vascular disrupting agent, which we feel will work very well with a TGF-beta antisense molecule. Why antisense? Antisense to us is the next generation platform. We all have been working on small molecule. We all know that smaller molecule is unpredictable in terms of its non-specific tox. The small molecule, you can change the backbone a little bit and you incur different toxicity that right now we're not able to predict. But the small molecule is cheap to manufacture, and so that has always been the mainstay of drug development. But then monoclonal antibody taken over since 1920s. There, the backbone is constant, so you're really not dealing with backbone toxicity. The target can be determined pretty precisely from the binding site on the monoclonal antibody, and the binding site can also be modified very readily in vitro, so that you can change binding affinity and all that. So it becomes a strong, robust platform for drug development. However, the cost of making a monoclonal antibody is high, and also to generate cell line that can make the monoclonal all the way to manufacturing is a slow and laborious process. Now, with that, we are moving on to antisense. Antisense been developing since the 1990s, and there's quite a few companies that's working on antisense with at least 6 or 7 drug approval on the market. Now, this has get a tremendous boost by the DNA RNA vaccine against COVID. However, the platform has always been there as a highly robust platform where the backbone is known. Therefore, the backbone toxicity is not something that we normally worry about unless we go in and modify the molecule, the backbone. Of course, there's a lot of modification in the backbone. So every backbone that's been modified, we have different toxicity. But once you settle on a single backbone, which what we did is we're using the phosphodiol backbone for all of our molecules. So we know the toxicity pretty well from the backbone. All you have to do is change the sequence and you would have a new drug targeting a new protein. And therefore, development can be fairly fast. There are several cases before COVID-19 where a drug from concept to first in man took less than 1 year. And with RNA vaccine and DNA vaccine for COVID, you can see that it can be greatly accelerated. Now, our drug, OT-101, is antisense against TGF-beta 2. It knocked down TGF-beta 2 by binding to the RNA sequence of TGF-beta 2. But because TGF-beta 2 regulates TGF-beta 1, knocking down TGF-beta 2, you knock down TGF-beta 1 also. The drug has been through over 6 clinical trials in oncology, and it has strong patent protection to 2037. We have orphan drug designation in US and EU. And manufacturing and processing is well understood. And we have enough drug to treat over 5,000 patients. So the antisense platform is robust in that sense that, like I said, it can go from gene identification to man in less than 1 year. And that's really how we can take forward drug development. And we all shouldn't stop thinking of drug development at 15, 20 years. Life is too short for that. Maybe with adoption of the antisense platform, we can accelerate all this. The single agent activity, the drug has activity against glioblastoma in G004 phase 2B trial, the 145 patients. About 100 of them actually went on to OT-101, and 45 was on a control arm with temozolomide. And here we took patients that have failed chemo, failed radiation, failed surgery, and recurred. These patients don't normally respond to further treatment, and yet on our drug, we're able to see significant clinical benefit in 60% of the patients from stable disease all the way to PR and some CR. Most interestingly, we see patients that have long-term survival, typical for immunotherapy. So that was confirmed in pancreatic cancer trial, the phase 1/2 clinical trial, where we took in 61 patients, 37 with pancreatic cancer, 19 with melanoma, and 5 with colorectal cancer. And again, we took patients that have failed Whipple surgery, we failed first line, second line, third line. These patients don't expect to survive longer than 3 months. And yet, most of them, we're able to stabilize the disease. And you have stable disease for, in some patients, extended out, in this one patient out to 40 months. And one patient where the tumor disappeared and that patient's still alive to now. This is very encouraging to us. And we are pushing forward to put the drug into a phase 3 trial. But because of COVID, we couldn't start the phase 3 program because patients would not be available to come in for infusion. Clinical trial got slowed down throughout the pandemic. But now that the pandemic is kind of waning, in most of the country, clinical trials are coming back. And we feel that this is the right time for us to initiate the phase 3 program for glioblastoma as well as pancreatic cancer. So we are in the process of moving that forward, and we're hoping to have first patient in by the year end or first quarter of next year. Among looking at the effect of OT101 in oncology. While we look at COVID, we found that OT101, by the nature of the inhibition of TGF beta, could have an important role in our arsenal against COVID-19, primarily because we think that TGF beta is the driver for COVID. And if you look at all the clinical programs that have failed against COVID-19, it underscored how little do we know about COVID-19 or any viral infections, such that we have thrown everything at this bug, and yet nothing really have been working except for dexamethasone. Even then, it's only working against severe COVID-19. So sometimes we need to not be too confident of our knowledge. We don't know what we don't know. And we need to really think out of the box when we face with a new agent, instead of trying to leverage on what we know and assume the new virus will behave as we predicted. So since day one, we have said that TGF beta is driving this virus replication. And there has been a lot of pushback in the field that TGF beta is not the main molecule. It is actually may cause more problem to COVID-19 if you shut down TGF beta. But since then, studies after studies have come out to show that that is not the case. In fact, there's a recent publication showing that in normal patients, you have no TGF beta in circulation, a very small amount. The moment you get infected with COVID-19, day 1 to day 10, as shown here, you have a tremendous surge in TGF beta, 50,000 to 100,000 units. And that continue until the patient recover completely from the virus. Now, what we have shown early on is that TGF beta drive the replication of the virus by the fact that when the virus infects the cells, it upregulates TGF beta, and then the TGF beta lock the cell cycle and allow the virus to replicate more. So that is essentially create a positive feedback loop that constantly churn out TGF beta. So you have this surge in TGF beta, which then will drive the pathology of COVID. But more importantly, the level of TGF beta in circulation is so high that any kind of damage to the organs, such as lung, liver, kidney, all those going to be covered over by scars. And this scarring of the organs is actually what you see in the post-COVID symptoms. And in fact, if you look at patients who recover from even mild COVID, those patients now have predisposition to subsequent events, whether it's pulmonary or kidney or cardiac. These patients have up to 4 to 5-fold higher predisposition to these subsequent events. So that's a big thing that we have to deal with, and yet the field is not focusing on that. So we're all focusing on knocking down the virus and whatnot, but we're not focusing on what going to happen when the organ is scarred and you have left with deficiency in organ function after the virus replication. So we put our OT101 into the clinical program in Brazil and Latin America, Peru, Argentina. And I must tell you that a clinical trial in a pandemic situation, as a clinical person, we have failed this pandemic because for every piece of document, that 500 other people have to review it, regulatory body have to review it. So it's no wonder that we haven't been able to develop a drug because it took us 1 year to run this trial of 30 patients or so to look at how TGF-beta can be blocked with OT101. And so that structurally, the clinical system need to be revamped so that clinical trial can match the potential of antisense. So if we can say we can go from concept to first in man in 1 year, we can't step back and say it will take another 15 years before you get the drug approved. That is not where the field should be going. We should be saying it take 1 year to go from concept to first in man. We did all the animal study, allow the safety to go in, and we should be able to go from phase 1 to approval within 2 to 3 years instead of the 15 years that we all talking about. In any case, what we have found is in Latin America, while we were conducting this trial, the virus has evolved significantly. When we first started, you have the usual COVID symptoms, loss of smell, take about 7 days to have the symptoms. By the time we close down the trial, we are looking at the virus that will hit the patient. The patient will die within 3 to 4 days on admittance. Whether they have loss of smell or not is irrelevant. It has evolved to not the traditional first COVID-19, but more like the flu-like symptom, but the symptom was very severe. And when one person is infected, the entire household is infected. So because of that, the entire uh, hospital system in Latin America is overwhelmed. We have patients who no longer qualify to go on to ICU because there's not enough ICU bed or even oxygen. For example, in Peru, we're in a situation there when a patient is greater than 30 years old, they're not going to have ICU bed. The ICU bed will not be reserved for anyone that is greater than 30 years old. So that's telling you a lot. In the US, we wouldn't have that problem. But in Latin America, with the medical system being overwhelmed, that's what happened. And even in the US during our heyday, when the virus was at its peak, some of these issues pop up is that the rationing of care, and also you dealing with the virus, you run into a situation where if there's no infection, then you don't have patients to go into your trial. But when the surge is happening, then all the clinical team are on the front line treating patients, and they can't do clinical research. So that's the other part that was slowing down the trial because the system is simply overwhelmed. In any case, that was Latin America. That trial was conducted by together by Covance. And that's the other problem with a large CRO is sometimes they don't move as quickly as you and I, a small investigator, a small CRO can move faster. Individual investigator, individual physician probably can move a lot faster than a CRO. In any case, we realized that there's no way that we can develop a drug fast enough to deal with this pandemic, given the traditional route of drug development. So what we did, we screened several thousand of compounds to look for one that has TGF-beta inhibition to match that of OT101 and to confirm that that also suppressed virus replication. And we found that artemisinin, an old malaria drug, coming from Artemisia plant, has strong TGF-beta inhibition. And when we tested in vitro in cell-based assay, we were able to show that it suppressed the virus replication. So we took that drug, uh, and since it's already available on the market, we're able to put it into a clinical trial fairly quickly. And we completed the RT19 trial in India with 120 patients. The data came out positive, consistent with our proposal that TGF-beta is important. And with that, it was adopted by Dr. K.K. Agarwal at Heart Care Foundation, and this is his in his protocol. For him, he would implement Pulmoheal when the patient is in cold 3, severe illness or comorbidity or high risk, with fever and CRP greater than 10 or rising. And he would add in Pulmoheal, which is artemisinin, marketed in India, in combination with dexamethasone. In our protocol, the RT19 protocol, we took patients with mild, moderate COVID without oxygen support, and we add in artemisinin on top of standard of care. And standard of care at the time in India was everything, including the kitchen sink, but it varied from site to site. One side is heavy on remdesivir, another side is heavy on dexamethasone, another side heavy on doxycycline, ivermectin. So that gave us an opportunity to evaluate the effectiveness of the drug across multiple standard of care. And what we have found is that in almost all cases, patients who went on to artemisinin recover faster. So that's consistent with our hypothesis. But most important is that if you look at their lung function throughout the monitoring period of 28 days, all patients who on artemisinin have fully recovered their O2 sat and their respiratory rate, whereas standard of care did not, which is consistent with what we see now that COVID-19, even with mild and moderate COVID, you don't really fully recover your respiratory function. So with that, our collaborator in Iran ran the artemisinin in combination with a couple other vitamins and herbal supplements against standard of care, which is antiviral, azithromycin, HCQ. And in this trial, consistent with our trial, they were able to show that patients clear rapidly on artemisinin their pneumonia. So here is a pneumonia before treatment, and you can see it's fairly extensive. It cleared very quickly after artemisinin treatment. But what's important is that O2 saturation fully recovered with artemisinin arm, but not with standard of care arm. So right now, this COVID protocol is approved in Iran, and so far they have gone on and treated over 5,000 patients. So summarizing our experience with artemisinin and COVID, this is the treatment schema that we have right now in India. If people are suspected of COVID or have respiratory symptoms, they go on to artemisinin once daily for 5 days while they get tested for COVID. And if they are positive for COVID, and we would recommend that they go on to standard care plus artemisinin, or if they have progressed to requiring oxygen, then they would use dexamethasone together with artemisinin. Or in situation where resources are not available, and people cannot afford the medication, then they can simply stay on artemisinin alone. And we've been providing artemisinin to people for free of charge to help combat this pandemic, along with other NGOs in India. our strategy in India is that there's a massive fire after COVID when it was at its peak. There were limited resources, so you don't want to just spread out your resource throughout and diluted the effort. Instead of that, we had to target our effort, and we deployed the artemisinin into one hotspot and to establish a containment zone. We treat family instead of individual because the virus had mutated in our watch, and that it had gone from infecting one person and not affecting anybody else to once one person is infected, the whole household is infected. So our um, mantra is to treat town, village one by one to establish a containment zone. So that so far been fairly effective in the NGO activity that we collaborate with in India. And we hope to move forward with this project to Southeast Asia. Right now, we in talk to deploy that in Philippines and hopefully in Indonesia when where the next wave is coming along as well. And we're also looking to see how we can deploy in Africa. But right now, the COVID has spread out of India and it's affecting Southeast Asia the most, but it's also going to really cause a significant spike in Africa. With that, I'd like to close and open up for a question if there's any.
Yep. Thank you for your. We are so glad that you could hear and today and share your insight. And I can see that we have two questions. In this moment. Yeah, I can see now. The first question is, what are the backbone of the antisense and what kind of nucleotides modification are used? So this is the first question.
Well, we use a phosphatidylate backbone because the DNA by itself will degrade it in circulation very quickly. We have a lot of nucleases in circulation that protect us from viral infections. So those nucleases will chew up the DNA fairly rapidly without formulation. And so we modify the backbone with phosphatidylate backbone instead of the regular deoxy backbone. And the second question is really what is the PK of the antisense? Now, antisense PK characterized by a very rapid distribution into tissues. So it have a very rapid distribution phase and a slow clearance phase. Clearance is by the kidney, and part of it, most of it by kidney, a little bit by liver. You have clearance is slower, but you have to keep in mind that distribution is very fast into the underlying tissues. So this is where the antisense fail early on against liquid tumor, because I know a lot of people say there was a big blow up with an antisense against BCL, and that didn't work. But that because the people who were developing the antisense were going after liquid tumor. And in liquid tumor, it's not going to work because the moment you inject it and the antisense, it immediately distributes to the underlying tissue. So there's just not enough of the drug to have an impact on the liquid tumor. In our experience, the drug distributes very rapidly and get into solid tumor. And in fact, if you have a liver lesions, those will be impacted very quickly with the drug. Whereas if you're dealing with circulating tumor or a liquid tumor, you're not going to have an opportunity to have that effect.
Thank you very much for addressing this question. I would have one question about treatment, the COVID treatment. Have you already identified other competitors who also develop treatment targeting with the TGF beta to treat as a approach to treat COVID-19?
We never really meant to go into COVID. We knew that TGF beta was important, so we tested our OT101 and other molecule against TGF beta, and we shown it to be active, and we released the data back when the pandemic started. The hope was that, okay, well, we gave you the data, just go do what you do best, and we go back to our oncology side. But the uptake was slow, so we are forced to conduct our own trial to convince people that it's actually working. But I think now that our data coming out and other people data coming out, matching our result, the field is changing to adapt to that. And plus, we have so many failure as therapeutic against the drug. I think that's also changing. But also the pandemic is waning. So I think most company like ourselves is in a situation where we say, well, this was important a year ago, but it's not so important now. As a company, we can't chase after a virus that is disappearing. So we're probably moving forward to oncology and put more effort into it while the study winding down in COVID.
Thank you. It looks like we've covered all of our questions. So I think that we can finish, or maybe you would like to, because we have 10 minutes, so maybe you would like to comment or cover other issue which you didn't deal in your presentation.
No, I think what we all have lived through this pandemic, and I hope that we all adapt to it and move forward with a different paradigm. Like clinical trials shouldn't take forever to get done. I think we all get complacent with the fact that that's just how things are, and you run the clinical trial years and years, and by the time you finish the phase 3, the original developer is probably dead by that time. So I think we should learn from this pandemic and move advanced clinical development out from its current status and try to move it quickly. And I think that's require collaboration between regulatory body as well as academic and physician and companies. But I think a lot of this thing is clinicians have shown to be very effective at putting drug into patient and showing that whether it's going to work against COVID or not. I think that's the important lesson to take away from this, is that a lot of the clinical development need to go back to where the clinician who can really drive it. Okay. Thank you.
Okay, one moment. I will check once again the asking box. Yeah. It was the question is it was the same question in my mind. How was antisense administered, oral or intravenously, and what dose? Yeah, I also was curious about the route of treatment.
The drug, the antisense were administered intravenous. Now we have two different approaches to deliver the drug. One is intratumoral, which is for the glioblastoma. That we directly administered the drug through a catheter to the tumor. And you don't need a whole lot of drug because you're essentially putting the drug right where it's supposed to be. Now, if you're dealing with tumor that easily accessible by intravenous route, then that's how we're going to deal with it. The pancreatic cancer, melanoma, colorectal, those we deliver the drug intravenously. So the dose is higher. Our safe dose was roughly about 300 milligram per meter square, but our effective dose was 140 milligram per meter square. So we chose 140 milligram per meter square, given the fact that we could never hit a toxic dose, or we could never see toxicity as far as we go in terms of the dose. So yeah, we chose the lower dose because it's already showing you efficacy, but I don't think there's an upper limit.
Okay. Thank you very much for addressing the last question. And as I mentioned now, I would like to introduce our next valuable speaker. Now, Professor Pan will present. Professor Pan has long studied the molecular and cellular mechanism of opioid actions that are mediated through mu opioid receptor splice variant using multidisciplinary approaches. These studies provide the foundation for developing novel analgesic drugs to treat pain devoid of many side effects associated with traditional opioids. Dr. Pan is a co-scientific founder at Sparian Biosciences, a company devoted to the development of safer alternatives of opioid drugs. And today, the title of his presentation